The goal of this symposium is to stimulate, inter-campus, interdisciplinary, interprogrammatic collaborations. The symposium is being held jointly with NORC, and I can see Paul McLean's back on, who's the director of NORC. We're hoping to have a lot more interest with them in the future. So, there's an RFA associated with this program. It should be on your invite, so you should have information on that. And again, one other thing I'm going to say. Anyway, I'll go just while I'm losing my mind here. I'm going to go and introduce the first speaker. So the first speaker is going to be Angelo D'Alessandro from my department, and he directs the metabolomics component of the mass spectrometry shared resource at the Cancer Center, one of our new shared resources. And he's going to talk about uncovering cancer vulnerabilities using metabolomics. And so, Angelo, you go ahead and share your screen and take it away.
Can you see my screen now?
Yes.
Thanks so much for the introduction, James. And I hope my internet will not be jumpy, as I had some issues in the last few days. They're literally working outside the house. So today I'm going to talk about uncovering cancer vulnerabilities using metabolomics, and specifically we'll focus on acute myeloid leukemia stem cells. So the presentation, I will not try to pretend to know anything about leukemia more than many of the attendees of this presentation. So just take this as an example. If you are not familiar with our lab, my lab personally is located on the 9th floor of RC1 South, and the core and all the fancy tools and instruments are on the 1st floor of the same RC1 South building. And we offer a series of services that also include proteomics. I can share this slide with you if you're interested in this, or feel free to reach out to me in private for any additional information. A lot of the things I'm going to be showing today, and I apologize to Harry and Brett down here, come from collaborative work of my lab with the Jordan lab and James' lab, James Segrevi's lab. And kudos to them for all the cool biology and animal experiments and human clinical trials that I will be showing today. Just for those of you who are unfamiliar with acute myeloid leukemia, for the sake of this presentation, it will suffice to say that it's a cancer of bone marrow-derived blood cells in which abnormal cells build up in bone marrow and blood and crowd out normal cells, blocking the proper development. Unfortunately, the prognosis is still dire. 5-year survival rate in adults is approximately 24%, which drops even further to 5% for individuals over the age of 60. And despite improvements over the last few years, I should say, and I will show some of them in this presentation, over the last several decades, most of the treatments have resulted in very high toxicity, and prognosis has improved much in this disease. So, and I'm gonna be moving this bar down here. The classic view for most cancer research is that you have, like, in this case, hematopoietic stem cells, and they acquire some mutation that favors their proliferation, making them leukemia stem cells. And these leukemia stem cells end up proliferating, resulting in the formation of these leukemic blasts that, as I was telling you, crowd the blood and the bone marrow and alter the proper development of the other hematopoietic stem cells. If we talk about mutation in acute myeloid leukemia, one common mutation is several mutations to the receptor tyrosine kinase FLT3, some of which are common and as high as in frequent as in 30% of all AML. And treatments have been developed over the years to target some of these mutations. I'm showing here, for example, AC220 to target some specific ITD mutation for FLT3. And if you use specific inhibitors of these receptor tyrosine kinase specifically for one of these mutations, AC220 in leukemia cancer cell lines, what you see is something that looks like this. So you have a lot of metabolites here in unreadable fonts over here. You go from red to blue, meaning that a lot of it is the cancer cells here are obliterated literally by the treatment with AC220. So what's the problem here? The problem is that unfortunately this conventional chemotherapy that literally obliterate the leukemic blast fails to actually target the leukemic stem cells that inevitably end up relapsing. So can we specifically identify something that is unique to these leukemia stem cells that can be targeted for eradication of the disease in long term and avoiding the relapse of the disease? I'm borrowing concepts here from Dr. Jordan in the early 2000s. He came up with this concept, a very elegant concept of leukemia stem cells, in which he identified two populations, one of the highly proliferating leukemic blasts, in which if you look at these cells typically, you will see that these cells are very susceptible to reactive oxygen species, and they have high levels of reactive oxygen species. From now on, for simplicity, we refer to them as ROS high. And it's really helpful for me to see the head of James moving slightly every now and then because I know I'm still online. But on the other hand, you see the ROS low population, the leukemic stem cell, are characterized with lower levels of reactive oxygen species, suggesting they are more resistant to the oxygen stress in the first place. If you were to sort these cells, the ROS high and ROS low, and look at the metabolic phenotypes of these cells, you will see that the highly proliferating leukemic blasts are characterized by higher glycolytic rates, while the leukemic stem cells, the ROS low population, is actually more reliant on 3-carboxylic acid cycle, mitochondrial metabolism. So here is where our lab came into play, specifically in collaboration with the Jordan Lab and Polya and Courtney Jones now at University of Toronto. We identified that leukemic stem cells have significantly higher levels of all amino acids. Specifically here I'm showing highlights of glutamate and cysteine, which are much higher in the ROS low leukemic stem cell population. And these metabolites in particular are precursor to glutathione. Reduced glutathione is the main antioxidant, hydrophilic antioxidant in the cell. This is relevant in that we can do, for example, tracing experiments with 13C, 15N labeled glutamine, so stabilized labeled tracers, in this case ex vivo, in vitro, in this case with MOM13 leukemia cell lines. And we can use, for example, AC220 and identify AC220 as an actual suppressant of glutamine uptake, but then we can use also inhibitors of other enzymes downstream, such as, for example, CB839, which inhibits glutamine conversion to glutamate by glutaminase activity. And this very glutamate can enter the TC cycle in the form of ketoglutarate or be used as a substrate to generate reduced glutathione, which would explain why the cells that resist the AC220 treatment and relapse are characterized by, tend to be characterized by lower reactive oxygen species. I anticipated this slide. If we do this tracing experiment and sort beforehand, before treating with AC220, the ROS low versus ROS high, you will see that the ROS low leukemic stem cell population are characterized by a faster rate of glutathione synthesis in the face of AC220 treatment. Additionally, these ROS low cells undergo transcriptional reprogramming to the extent that several genes that I'm showing here in the upper right panel, such as, for example, BCL2, are upregulated in the leukemia stem cells. It is relevant in that, for example, BCL2 counteracts apoptosis, especially the intrinsic mitochondrial pathway that is activated by PV3-dependent cascades. So the question here is, can we target both the metabolic vulnerability of dependency on, for example, glutamine to, or amino acid metabolism to fuel the TCA cycle and counteract oxygen stress, at the same time inhibit the anti-apoptotic pathway that is driven by BCL2? And Dr. Polly and Jordan published a series of very interesting papers that I'm sure most of you are familiar with, in which they use a combination of BCL2 inhibitor, venetoclax, plus azacitidine, which is a DNA methyltransferase inhibitor, the rationale being there that a low dose is azacitidine is an inhibitor of DNA methyltransferase, so it's the DNA methyltransferase dependent methylation of DNA results in silencing of pro-apoptotic pathways. At high doses, the same azacitidine intercalates in DNA and RNA. It's actually cytotoxic itself. So the combination of venetoclax and azacitidine actually was extremely effective in decreasing, red to blue, the levels of all amino acids that are indeed higher in the leukemic stem cells before the treatment. However, we can keep the cells proliferating in the phase of veneza treatment by incubating them ex vivo with 10 times the normal concentration, supraphysiological concentration of amino acids. So to further elucidate what the mechanism could be there is, I showed you that these cells rely on amino acids, for example, for the synthesis of glutathione, and in the phase of veneza treatment, red versus black here, these cells decrease their levels of intracellular glutathione, and in the phase of veneza treatment, you actually see an accumulation of succinate, the substrate for complex 2 mitochondria. Why is that? Specifically, the glutathione in these leukemic stem cells is used to glutathionate complex 2 mitochondria and keep it active. However, when glutamine uptake and consumption is inhibited by veneza combination, by constraining glutamine availability and constraining glutamine metabolism, the result is a depletion of reduced glutathione, an increase in the overall reactive oxygen species in the cell, and an inhibition of complex 2 activity by lack of the protective glutathione effect on complex 2. This phenotype can be rescued by administering, treating the cells with glutathione, reduced glutathione in the phase of veneza treatment. So the really good news is that when you go to actual clinical patients in the clinics, this treatment seems to be very effective. In this beautiful Nature Medicine paper, Dr. Pollia demonstrated that approximately 33 patients enrolled in this trial, over 80% of them were cancer-free, leukemia-free, over 3 years from the beginning of the treatment. If you do, as they did, perform single-cell RNA sequencing in this subject, what they could see is that the leukemic blast completely disappeared here after 4 days of treatment, and the leukemic blast population did not relapse. So this was all over the news. It was very promising, and it is still a treatment that works really well in approximately 70% of patients. Unfortunately, in approximately 30% of patients, there were most of which were previously on other regimens, we still observe a relapse. So the question here is, can we identify metabolic vulnerabilities in these additionally relapsing cells that are resistant to veneza treatment? So in collaboration with, again, the Jordan Lab, Dr. Brett Stevens and Shashan Pai and Courtney Jones, we and a very talented student, a graduate student in the lab, Rachel Cole-Pale, performed metabolomics analysis of these relapsing cells. So they had the leukemia cells from patients, a diagnosis and relapse, and we performed metabolomics analysis to identify that in a single one of those subjects, we could observe significant increases in fatty acids, in the levels of free fatty acids in the sample cell relapse. We could perform 13C palmitate tracing experiment, in which each one of the 16 carbon atoms of palmitate is labeled, and then look at the ROS low versus ROS high population of leukemic stem cells versus leukemic blast, and identify that in response to veneza treatment or in response to amino acid starvation in the media, the low ROS leukemic stem cell population has the flexibility to switch to consuming palmitate, consuming fatty acids in the face of amino acid starvation. And the oxygen consumption rate in these relapsing cells, even in the face of veneza treatment, venetoclax plus azacitidine treatment, is still consistent with what you would observe in the cells treated with vehicle alone, suggesting that veneza is insufficient to suppress oxygen consumption rate in these cells, as it is in other cells at diagnosis. If you perform again this palmitate tracing experiment, you can actually look at trace the incorporation of available carbon atom from palmitate into TC cycle intermediate and see that these relapsing cells incorporate carbon atom derived from palmitate at a much higher rate than cells at diagnosis. To the extent that if you add to the combination veneza, etomoxer, a currently implemented transferase inhibitor, even though this is controversial in the field, it could also be considered a complex 1 inhibitor, you see that the percentage of viable relapsing cells significantly decreases. These cells are so apparently addicted to palmitate that you can actually incubate them with uniformly labeled 13C6 glucose, so glucose that is labeled each one of the 6 carbon atoms, and trace this carbon atom incorporation into fatty acids, but not saturated fatty acids, to a lesser extent monounsaturated fatty acid. Mostly, these carbon atoms end up accumulating in polyunsaturated fatty acids. So again, if you look at lipidomics, more complex lipidomics analysis, the saturated lipids don't increase much in the relapsing population, here in red versus blue, but all the unsaturated lipids we could detect in this analysis were significantly upregulated, increased in the relapsing cells compared to the diagnosis cells. And here I'm showing again the relapsing in red versus diagnosis in blue for the several polyunsaturated and monounsaturated fatty acids in phosphatidylcholine and phosphatidylphenolamines, which are the most abundant phospholipids in the cell. So what is driving this unsaturation increase, not just in fatty acids, but in the degree of unsaturation of these fatty acids? The pathway that regulates fatty acid desaturation relies on these enzymes that are called fatty acid desaturases. You see FATS2 and FATS1 almost at the top of this pathway. And if you look at the cancer genome atlas, and we need to thank Dr. Shanshampai for this analysis, the subjects expressing the highest levels of different fatty acid desaturases have the worst, poorest prognosis in acute myeloid leukemia. So what we decided to do here is to combine veneza with the shRNA treatment towards fatty acid desaturases. I'm showing here, for simplicity, just the shRNA towards the most upstream of the fatty acid desaturases, FATS2. And you see that the percentage of viable cells increases significantly when veneza treatment is combined with fatty acid desaturase downregulation at the post-translational level. So the take-home message here is that we could identify through a combination of metabolomics analysis and serial intervention in these cells a metabolic vulnerability that pertains to the activity of fatty acid desaturases. We believe to to recycle NADH in the face of a dysfunctional mitochondrial metabolism in response to veneza treatment. And in so doing, this fatty acid desaturase activity recycles NADH back to NAD+ and keeps energy metabolism going and TCA cycle going for the purposes of the cells. Targeting this fatty acid desaturase could result in a potential therapeutic intervention in these relapsing cells in patients who relapse upon veneza treatment. And with that, I would like to thank everybody in the lab and the Jordan lab and James Agaro's lab and other collaborators in the field of space of acute myeloid leukemia. I would be happy to take any question. Thank you.
Great, that was great, Angelo. You were right on time too. So open up the floor for questions.
Hi, Angelo. This is Sid Mitra from Pediatrics. Can you guys hear me?
I can, thank you.
Yeah, that was very interesting talk and very nice work. One of the things that I'm really been interested in, or rather something that has always inhibited me from going into metabolomics, has been I am never quite sure. So what are the cell numbers here that we are talking about in terms of, like, what is the minimum number of cells that you really need to get a decent signal when you're looking at metabolomic analysis? And almost a follow-up to that question is that I'm assuming a cellular metabolomics is far more sensitive or time sensitive compared to, let's say, even single cell RNA sequencing. So do you know how much change really occurs if you put a tissue through flow cytometry or something, or sort out the cells and then start doing metabolomics? How much does that end up changing?
So one recommendation there is to keep the cells at 4 Celsius as quick and perform all these sorting as quickly as possible. So that's definitely a pertinent point. One caveat around that is usually to include, for example, a step of ex vivo incubation again with media, but that too introduces an additional step and potentially affects the quality of the analysis. In terms of the limitations in terms of cell counts, I'm showing here some preliminary data from a proposal we just submitted in collaboration with Dr. Marie-Dominique Filippi at University of Cincinnati and Jose Cancelas, University of Cincinnati, Oxford Blood Center. In this experiment in particular, they literally took the cells one by one under the microscope, and we performed metabolomics analysis on 320 cells. Drew some biologically meaningful conclusion that informed some intervention as a follow-up of these analysis. So the point here being that we have right now the technology to perform single-cell metabolomics with, for example, nano-HPLC mass spectrometry or capillary electrophoresis coupled to mass spectrometry. The reason why we haven't pushed towards that direction is because of the time constraints for this kind of analysis. The instruments are sensitive enough. The problem is that if you run a cell every 5 minutes, it will take you, 2-3 weeks worth of metabolomics analysis to have a meaningful number of cells to draw any meaningful conclusion on a larger population. But if you have a capacity to sort smaller subpopulation, we've been working, for example, in the context of non-malignant hematopoiesis, and specifically erythropoiesis, which is my main expertise on red blood cell biology and erythropoiesis. Then you can definitely go down in the low hundreds to few thousand cells. Ideally, if you have 100,000 cells, you make us happy. But depending on the biological question being asked, you can obviate with, for example, performing specifically targeted analysis that increase the sensitivity of the approach. So I have a less comprehensive analysis, but look at specifically 200 to 300 metabolites in a targeted fashion, rather than looking potentially of several tens of thousands of features, few thousands of named metabolite in more heterogeneous, larger population.
Great. We have time for one more question. If someone else has one out there.
I have one. It's Jennifer. Hey, I'm just wondering what you guys think. It might take a few people to chime in on this question, but it seems like in the solid tumors, like in carcinomas, it might be kind of a different situation, like a little bit flip-flopped, because we see that the more rare stem-like cells that can survive in unattached conditions, epithelial cells and from which carcinomas are derived, like the attached conditions. So they like to be on a basement membrane or in the primary tumor, and the ones that can live in the detached condition, so similar to the natural state for a cancer derived from a hematopoietic cell. Anyway, we see the opposite, where they have higher ROS, these rare stem-like cells in the carcinomas, but they're also making a lot of antioxidants to try to keep the levels of ROS from being cytotoxic. And I'm just wondering if this might be sort of a difference between the hematopoietic cancers and carcinomas.
Yeah, now finally you can see you. Switch to the gallery mode. That's a great point, and I don't want to talk about something that I don't know in detail, but if I am allowed to speculate in just for a few seconds. Your work, Jennifer, and your group's work on kynurenine as a potential immunomodulator metabolite is extremely relevant to this question, to this very question, in that the pathway that generate these metabolites that make these cells escape immune patrolling and immune recognition are stimulated by pro-oxidant stress and pro-inflammatory pathways. The very step that generates kynurenine by tryptophan oxidation by indole-2,3 dioxygenase concomitantly generates the active oxygen species or at least precursor in the form of hydrogen peroxide. So that, I think, may lie your point. Like when we do these experiments in a context that is evolved, that is separated from how the system react to that cell, so excluding, for example, the immune responses to the cells, that may bring us to a direction that is different from what you've observed. So my understanding is that these therapies, these regimens would actually be really effective in targeting the cells directly, but I don't know what they would do to the immune cells seeing these cancer cells derived from solid tumors.
Great discussion. We're going to have to move on to the next talk, just to keep on time. And let me pull it up. So the next speaker is going to be Dr. Mayumi Fujita, and she's in the Department of Dermatology, where she's a professor, and she'll be talking about the role of acetaldehyde and mitochondrial enzyme ALDH2 in melanoma. So Mayumi, if you want to go ahead and share your screen.
Sure, thank you. Oh, do you see it? I hope. Okay.
Yes.
Anyway, okay. Yeah, thank you, James, and also Cancer Center leaders for selecting our story. So my research team has studied tumor-induced inflammation and inflammasomes here, and also tumor heterogeneity and immune suppression. But today, I'm going to show you new data from a new project that we recently started. Oh, yeah, this one, okay. So the cancer that we study is still melanoma. So the skin cancer from melanocytes, pigment-producing cells in the epidermis. Okay, yeah. Okay. So this is one of the most common cancers, and the incidence of melanoma has increased every year. So we wonder, why does melanoma continue to increase? So there are many risk factors that have been already identified, including genetic factors, and also phenotypic factors, and environmental factors. Specifically, this UV is one of the major factors contributing to skin cancers. So we dermatologists have campaigned for the recommending sunscreen use and also UV protection. But we still see the increased number of melanoma and skin cancers. So are we doing the right job, or are we missing something? So we analyzed the map of melanoma incidence and also some risk factors. So this is the UV effect. So you can see that melanoma incidence and UV exposure do not seem to correlate very well, even though we combine skin color as another factor. Also, melanoma tumors do not always occur in sun-exposed areas. So are there any environmental risk factors? We do know that pregnancies and immunosuppressive drugs can be the risk factors for melanomagenesis. But not everybody is pregnant, and also not everybody takes immunosuppressive drugs. So are there any common factors? So obesity could be one of them. Smoking could be also one of them, another one. And how about diet? So recent data and studies show a strong correlation between alcohol intake and melanoma incidence from meta-analysis and other studies. So we use these maps again. So this is the obesity prevalence. smoking, and alcohol. So you can see some correlation between melanoma incidence and some of the risk factors here. But I hope you can appreciate that the patterns from melanoma prevalence is very similar to that from melanoma incidence. We also use the US map to analyze and assess the melanoma incidence, obesity, and smoking, and alcohol consumption. And you can see some similar trend here, although they are not completely matching. So we wonder how alcohol can induce carcinogenesis. To answer this question, we have to understand how alcohol or ethanol is metabolized. So this ethanol is oxidized to acetaldehyde by ADH, alcohol dehydrogenase. And this is the carcinogen. And this carcinogen is oxidized to non-toxic acetic acid by ALDH2, aldehyde dehydrogenase 2. So we thought probably some of the enzymes could be involved in carcinogenesis. And we use TCGA dataset to analyze the melanoma gene expression for these 5 genes. And we did see the clear, beautiful separation from high expressors of ALDH2 and low expressors of ALDH2. We also studied the ALDH2 expression levels, and we found that most melanomas do downregulate ALDH2 expression. So this also means that even the high expressors with better prognosis already have the downregulated ALDH2. And when ALDH2 is downregulated, you can understand that acetaldehyde could be accumulated. And this is the carcinogen. So how can ALDH2 be downregulated or inactivated? Well, there are many mechanisms to explain this, the genetic levels and also the protein levels, because this is also the mitochondrial protein. And specifically, environmental factors, many factors, such as alcohol, high-fat diet, smoking, drugs, and mitochondrial dysfunction, and oxidative stress can induce post-translational modification of ALDH2 to inactivate this ALDH2 enzyme. And when this is decreased, acetaldehyde is built up. And acetaldehyde is a carcinogen to damage DNA directly, and also modify proteins and induce oxidative stress, all of which can contribute to tumor initiation and progression. So you may think that, well, this would be an interesting study, but I do not drink alcohol, or I'm not a heavy drinker. But actually, even though you do not consume alcohol beverages, you are exposed to ethanol. The sources of ethanol include a variety of food, medications, microbiota, which can also cause old Blue-Murray syndrome, and also even the non-alcoholic drinks contain some alcohol. How about acetaldehyde? It is worse than ethanol. Most people would be exposed to acetaldehyde environmentally from fireplaces, wood stoves, coffee and fumes, and also occupationally, and smoking, even perfume or preservatives and flavoring agent, and of course microbiota and drinking and other metabolisms. So I hope you understand that we have the importance of studying this effect of ethanol and acetaldehyde in carcinogenesis. So to study this, we use ALDH2 knockout mice. So by using the ALDH2 knockout mice, we can assess the ethanol-induced acetaldehyde effect. So this is the experimental setup. So we had knockout and wild type and het. and we put ethanol into the drinking water because we wanted to study the chronic effect. And we did see a tremendous skin changes. So some of you who did not study or who do not use mice may not know which one is normal. But even though we use black 6 mice, their tail and paws should not have pigment. So this is normal. So it should be pinkish or grayish. And we did see the tremendous amount of hyperpigmentation. And this hyperpigmentation was dose dependent and also genotype dependent. Because acetaldehyde can damage DNA, we wanted to know whether that can also induce UV specific damage or just a general DNA damage. Then we wanted to study non-DNA changes. UV induces so many changes, but these two factors are very important. UV induces ROS directly to melanocytes, and UV can also induce and activate adenylate cyclase indirectly through alpha MSH production from keratinocytes. And these two factors are very important to activate so many pathways. So we studied this. And also before that, we searched some papers from the effect of the alcohol and acetaldehyde. And many people use hepatocyte liver cells. But we found that alcohol can directly activate adenylate cyclase. And of course, acetaldehyde and alcohol can induce ROS, particularly when LDH2 is impaired. So we treated human melanocytes by alcohol, and we did see the huge increase of adenylate cyclase activity. And this is the UV-induced activity. So this is indirect, not a direct effect. And alcohol can directly activate adenylate cyclase. We also studied ROS, and when human melanocytes were treated with alcohol, we could see the production of ROS. And when they were further treated with LH2 inhibitor, we did see the increase of ROS here. Then we wanted to see the phenotypes occurring after this change. And we found that ethanol and LH2 inhibitor can induce the elongation of the melanocyte dendrite, and also more melanin pigment production, and also the distribution change from central distribution to the tip. And these findings are very consistent with the finding that we see from activated melanocytes after UVB exposure. And we also did see in vivo that melanocyte numbers were increased. And you can also appreciate more melanin pigment production here. So we did see the UV specific DNA damage and increased melanin and melanocyte activation and increased melanocyte number. And I also want to show this one more time. Okay, so this is the last piece of the slide data. So I did say that melanocyte numbers were increased in LH2 knockout mice after ethanol treatment. But here, we also observe something else. So melanocytes, okay, they are located here, and they bind to, attached to the basement membrane. So they should be localized in the basal layer. But we did see something here. So when melanocytes are transformed, they go up, and also they proliferate laterally. So this initial phase is called radial growth phase melanoma. And this upward spreading is also called pagetoid spreading. And then after that, they invade vertically. So this could be the signs of early transformation or pre-malignant signs. So we wonder whether acetaldehyde could induce melanomagenesis similar to UV. So in order to answer this question, we first have to understand how UV can induce melanomagenesis. And many people use genetically engineered mouse models of melanoma. So this is the summary of the study from other people. So similar to other cancers. Melanoma need activation of oncogene pathways and inactivation of tumor suppressor pathways. So many people crossbreed these two. And also they add UVB or UVA sometimes to neonates, 3-day neonates. But we want to study ethanol-induced melanomagenesis. So we want to use adult mice. So there was only one paper about this. So we are going to use this mouse model. And then we plan to crossbreed with ALH2 knockout mice or use ALH2 inhibitor and treat with ethanol. So this is the future, yes, that's the last one. So this is a future grant proposal. So we want to study in vitro and in vivo effect of ethanol and acetaldehyde in BRAF mutant melanocytes. And even though we already started generating some data, we do need more data. And this should be proposed in the pilot grant. So we need expert of proteomics and metabolomics and mitochondrial function, DNA repairs, or anything that you think would be helpful. And this is the last slide. So these are the people who have contributed to the study. So Takeshi and Ziwi studied ALH2 in melanocytes and melanoma. So most of the data that I generated today are from Takeshi. Thank you so much for your attention.
Great. Thank you so much, Manami. That's really fascinating.
Oh,
So
thank you.
Do you think there's, I'll start with one. Do you think there's expansion of the melanocyte niche, or do you think that they just become niche independent? Because it seems like, and that doesn't seem to be just simply due to mutations because it's so widespread across the mouse. So if it was a stochastic mutational event, I would expect it to be rare, not so widespread.
Right. So actually, what we saw is just a skin pigmentation. Okay. So
Yeah.
melanocytes were activated, but we did not observe melanomagenesis yet. Melanoma. Okay.
Right. But you saw the expansion of the melanocytes to higher strata away from the basement membrane, which suggests that they're no longer dependent on their normal niche.
But actually, it was also not strong enough. So yeah, when we stopped alcohol, they gradually reversed to the original state, not completely. Right. Okay.
Yeah, no, it's
but anyway, so yeah, so we think probably oncogene would be necessary.
Yeah. And Lily Nguyen just had a question. Do these epidermal? Oh, I just lost it. I got to go to the questions now. Oh. Do you see it there? Let me pull it up.
What? Where?
Lily, if you want to just ask your question, feel free to.
Yeah, so I just asked if these epidermal melanocytes originate from the same niche as the hair follicle melanocytes.
Well, yeah. So, of course, yeah. So epidermal melanocyte. Actually, this is we're talking about mouse here. So, but anyway, so mouse skin is a little bit different from human skin. But of course, most of the melanocyte, epidermal melanocytes, are from the hair follicle bulge. But also, some data also suggests that dermal melanocyte can also migrate to the epidermis.
Okay, thanks.
We have time for one more question if there's another one out there.
James, Patricia put one in the chat. Do mice metabolize alcohol like humans?
Oh, I think so. Yeah, there are so many mouse data. I'm new to the alcohol study, but many people have used alcohol in mice, and at least this ethanol and acetaldehyde, this pathway, and also the involvement of CYP2, E1, and also ADH, ALH2 seems to be almost identical, but it's possible that there might be something different.
This is Liz Kovacs. The alcohol is metabolized more rapidly in mice and rats than in humans.
Did everyone get that from Liz?
More rapidly.
Yeah.
Mm-hmm.
Great. Fantastic. So basically, Mayumi, what you're telling us, despite the fact that we're all stuck in quarantine, we can't drink. This is great.
Yeah, so yeah, we can drink. Yeah, so as long as we have very good ADH2, okay, so we just have to keep the ADH2.
Okay.
And when ADH2 is downregulated, then we have problem. Okay? And then high fat diet or oxidative stress can downregulate or inactivate ADH2. That's the one that we want to avoid.
Right.
Or of course, too much of the alcohol can also cause the problem.
Right. Absolutely.
Yes.
All right. Oh, actually, we do have one. So I'll ask one quick thing because we have time for one. We said there are genetic factors, right? So, you know, the LDH2 and 1 are, you know, highly variable around the world, their expression and activities. And do you know how that might be influencing?
I'm sorry, so are you talking about ADH1 or ADH2?
I thought there were polymorphisms in LDH2 as well.
Yes.
That was very polymorphic.
Yes. So actually, many Asians have polymorphism of ADH2. So, you know, they have redness after drinking alcohol. So they don't have good ADH2 activity. So
you might think they'd be more susceptible to this melanogenesis.
Well, that's also very interesting. We do have some clinical data correlating with ADH2 phenotype, genotype, and skin pigmentation. And also we started collaborating with the Japanese cancer center, and they are going to check all melanoma patients and their genotype, ADH2 polymorphism, because 40% of them have ADH2 polymorphism and 60% is the normal one. So it's very easy to study. And also actually there are some more interesting data. ADH2 of course is not just for melanoma. Okay, so other cancers also have this pattern, and also immune cells also have the effect from ADH2 knockdown, and immune response is different. Immunotherapy response is different in cancer patients.
Great.
There are some clinical data, that we have. Mm-hmm.
Yeah, fantastic, thank you. And we better move on to the next one. And just as a reminder, everyone, please actually, instead of putting your questions into chat, we may or may not notice them, go ahead and just ask them out. One of the goals here is to try to make this seem a little bit more live. And in fact, to do that, I'll actually show myself when I introduce the next person. So the next speaker is going to be Dr. Natalie Surkova. She's the director of our Animal Imaging Shared Resource, and she's a professor in radiology. And she'll be talking about NMR-based metabolomics of radiation and TMZ-induced inflammation in glioma models. So Natalie, if you want to take it away.
Yes, thanks, James. Can you hear me? Hello?
Yes,
Yeah.
we can.
Oh, perfect. I'm going to share my screen now. Okay. Okay. So we're going to talk about NMR-based metabolomics on temozolomide and radiation-induced inflammation, or so-called pseudoprogression in mouse glioma models. And I'll give you a brief background. What is the clinical problem for this study is, also our hypothesis, and introduce you to our NMR metabolomics approach for in vitro GBM cells. GBM stands for glioblastoma. Also, we'll talk about our data on NMR metabolomics in mouse GBM models. And since obviously I cannot give a talk without giving you some nice MRI images on mouse brain, we will also talk about iron oxide enhanced T2-weighted MRI for the inflammation in vivo, and discuss the most important findings and also the future studies. So the background is the GBM is obviously one of the deadliest tumors which humans can experience, and the treatment include resection and followed by chemotherapy with TMZ, temozolomide, and radiation. The problem with the TMZ and RT treatment after the resection is that on the diagnostic MRI, it can cause so-called pseudo-progression, which not true progression, but it is a phenomenon which is caused by local inflammation induced by radiation treatment and temozolomide. And as of today, by using the typical radiological approaches, we cannot distinguish pseudo-progression, which can actually extend up to 6 months after completion RT and TMZ from true progression. And obviously it's very important to know, since the survival rate and survival time is so short for glioblastoma patients, we need to know as soon as possible, are we dealing with the true progression after treatment or pseudo-progression? So this is how pseudo-progression looks on the clinical MRI. This is a diagnostic MRI scan obtained prior to the treatment to show the GBM and the area of enhancement on the brain MRI. And 1 month after radiation treatment with TMZ, we still see this persistent enhancement. And the question is it expanding glioma, or is actually pseudo-progression because of the inflammatory response to radiation and TMZ? And if you look here, actually I cannot see because I see, oh, it's okay. And if you look here, 12 months after radiation treatment, this enhanced area has resolved. So actually this response in 1 month was pseudo-progression, not true progression. So maybe PET can help us to distinguish pseudo-progression from true progression. However, unfortunately, using the conventional FDG PET, which uses radioactive glucose to depicture the area of cancer, we cannot see in the brain the GBM. The reason for this is that the normal brain in the background has a very high glucose uptake. So to see increased GBM uptake for glucose, since the majority of the tumor have increased glucose uptake, is unfortunately almost impossible in the brain due to the very high background. So our goal is to see if there's a new metabolic markers of GBM which can be translated in for their differences from GBM to pseudo-progression, and if we can translate those metabolic markers into the imaging approaches, so we can do actually non-invasive imaging with the metabolic biomarker or inflammation marker to distinguish true progression from pseudo-progression. So I will tell you a little bit more about the metabolic markers by metabolomics, but I also will tell you a little bit about the translation of those macrophage imaging and the specific metabolic biomarkers into the non-invasive imaging using MRI or PET. So let's just start with the methods. Angelo is our world-known expert in metabolomics using mass spectrometry. However, you can also do metabolomics using NMR. Actually, it's probably one of the oldest techniques to do metabolomics in human or animal tissues. So this is how NMR scanner or spectrometer looks like. And we do have a very thin capillaries filled with the tissue extracts, which we put inside of the scanner, and only after 5 to 10 minutes of the acquisition time for so-called proton NMR, you can see untargeted spectra, meaning you see all highly abundant metabolites in your sample. And proton NMR allows for global and quantitative profile of water-soluble and lipid metabolites, including amino acids, neurotransmitter, glucose, intermediates, lipids, and particularly important lactate. So from really 5 minutes of scan, you can get up to 200 quantitative endpoints for extracted blood cells, urine samples. So in the first part of our study, we used cell lines, and in particularly murine glioblastoma GL261 and human glioblastoma U251. The cells were treated with temozolomide irradiation, and the survival assay included tryptophan blue and clonogenic assay. And we also compared the changes in metabolism between the glioblastoma cell lines and primary mouse astrocytes to see what kind of the GBM metabolic markers we can distinguish and how they correspond to the treatment response to TMZ in radiation. And we did NMR metabolomics afterwards. So let's start with the survival assay where we're choosing our doses. TMZ alone does not really do much to the U251 or mouse glioblastoma cells. Radiation, on the other hand, in vitro, produces drastic decrease in cell viability. So we decided at this time point, I apologize, to use 30 micromolar TMZ treatment on our cells and approximately 3 gray of radiation in combination to see what is going on as a treatment response. Once again, we did want to kill the cells because we don't want to see the metabolic response to the killing the cells. We really want to see what happens when we targeted with the targeted inhibition of the tumor growth. And this is how the quantitative data sets of in NMR metabolomics look like. You can see that we can see aromatic amino acids, we can see polyols, glycine, taurine, we can see lysine, alanine, lactate, we can see many phospholipids and overall lipids. And once again, this is all quantitative, expressed as micromole per gram, milligram of the cells or biopsy. And when we look at the discriminative analysis, we can see there's a very nice discrimination in the metabolic profile based on those data sets between primary astrocytes and GL261, murine GBM, and astrocytes in human U251. So when we looked at the, and I have to make this move somewhere because I cannot see. Okay, perfect. This is better. So when we look at the metabolites which contribute to this group separation, we can see that lactate was increased in GBM up to 400% compared to the primary cells. The membrane phospholipids such as phosphatidylcholine were highly increased, and interestingly, the amino acids were highly increased between the normal and both GBM cells. So we went then to the mouse models, and we want to see if the same markers and treatment response will be seen in the mouse models. And we use wild type animals with flank or orthotopic GL261 GBM, nude mice with the human GBM orthotopic or flank, and we treated with them with TMZ and RT, and we looked for the NMR on the resected tumors after the tumors were confirmed with the MRI. And we also use pheromoxetol iron oxide nanoparticle, which are very specific for macrophage, to confirm that what we see after the treatment is truly macrophage-induced treatment response. So when we did the same thing with the, those big, large quantitative metabolic data sets on the healthy mouse brain, human xenografts treated with radiation and TMZ, mouse GBM treated with the TMZ, and also used for the untreated, for example, GBM, we saw that we see very nice clustering for the metabolic profiling. Now, I'm saying up here, I said that we actually called the pseudoprogression or inflammation in the human xenografts as a low inflammation, because obviously nude mice have slightly compromised response on producing macrophages to the treatment because they are just nude mice, right? And in the mouse isograft, we saw the high inflammation. So because obviously those are wild type, and this was confirmed also by by MRI. But for now, you just have to believe me that immune mice have low inflammation and wild-type mice have high inflammation. Excuse me. So when we asked what kind of metabolic markers are there to discriminate among all those groups, the highest metabolic pathway which came to distinguish among all of those groups were amino acids: methionine, tyrosine, and glycine. When we compared human treated xenografts versus mouse isografts, we saw that, yeah, there was some difference in lactate between those two high and low inflammatory groups. Glucose was almost unchanged between the groups. The phosphocholine, the membrane synthesis was a little slightly different. But once again, the highest differences between the low inflammation and high inflammation groups treated with the RT and TMZ were in amino acids. And interestingly, I just put here the amino acids which we saw as the differences between GBM and normal brain. Glutamine was not anymore significant between the low and high inflammation groups. So we also wanted to confirm that what we see, the differences between so-called low and high inflammation, are really in the presence of macrophages. And this is a human glioblastoma, so our low inflammation group. And if you add ferromoxytol, which are iron oxide nanoparticles, those iron oxide nanoparticles taken up by the macrophages only. Cancer cells don't care about iron oxide nanoparticles. And iron will actually drop your T2 relaxation time. So when we do pre-iron oxide versus post-iron oxide MRI in the human xenografts, we see only slight decrease in the T2 relaxation time, -5 milliseconds. And we saw also really not so many F480 cells, which are typical for macrophages on the flow cytometry. However, if we look at the wild type isograft GBM, treated with the RT and TMZ, pre and post injection of nanoparticles, we can see that our MRI looks much darker, which means we have a much higher decrease in the T2 relaxation time due to the presence of iron oxide, which were trapped in the macrophages and only macrophages, so -20 milliseconds. And here's the flow cytometry. And here's also Prussian blue for the iron oxide staging in our tumors. So we looked a little bit more in amino acid uptake using, once again, back ex vivo and using 13C substrates, 13C glutamine, 13C glycine, 13C methionine, and tyrosine, because those were highest VIP biomarkers between high and low inflammation groups. So really like true response versus inflammatory response after RT and TMZ. And we saw that primary cells, such as the primary astrocytes and brain slices, so primary brain cells, they don't really have high level of uptake for those amino acids. However, all brain tumor cells have high uptake of the C13 glycine, methionine, tyrosine, and glutamine. However, we also looked for the macrophages. And interestingly, primary macrophages love C13 glutamine and only glutamine. So how is it important to our study? If we want to see amino acids as a pathway to distinguish between true progression versus inflammation, pseudoprogression, then we can use amino acids which are increased in GBM. However, glutamine uptake is also increased in macrophages, so it will also be increased in inflammation. So if we want to distinguish pseudoprogression inflammation from true progression, yes, we can use methionine tracer, we can use tyrosine tracer and glycine tracer for the imaging, but we cannot use the glutamine for our future studies. And indeed, so this is a relatively recent study which came out from the UK and Germany, and they just showed that C11 methionine is increased in human low-grade gliomas. So they didn't do pseudo-progression versus true progression in GBM yet, but this is a very, very promising study because you can see that using C11 methionine uptake on the PET-CT in humans, you can see increased uptake in the gliomas compared to the normal brain. And I think this was my last slide, and I have 15 minutes here. So I just want to acknowledge all my team members who contributed to this work and my students, and we'll work very closely with the neuro-oncology team, Dr. Vibakar and specifically Dr. Angela Pierce. They helped us tremendously to do all those mouse models. And we also work very closely with Dr. Graner and Dr. Karam for the tumor biology and radiation treatment, respectively. Thank you.
Great, thanks, Natalie. Right on time. Do we have any questions?
So this is
everybody's talking now.
And a lot of work. And since we have collaborated for a long time, so I can appreciate the amount of effort goes to do this kind of work. When you're talking about your orthotopic xenograft data, I mean, this is like total cells, right? It's not that it's divided and pull the cell types out and look at how they are different between the, you know what I'm saying?
I think so. So you mean the in vivo studies when we're looking for the metabolism of the tumor?
Yeah.
Right. Yeah. So no, this is, well, I think as you said before, so one of the downside of the metabolomics, we do need a lot of cells, especially for NMR. So it would be almost impossible for me to do like single cell, metabolomics analysis. Maybe Angela can do this, but I definitely cannot do this. So this was, the tumor size was for the flank xenografts was about 150 milligram, as you know, is what I use. We always like to have. And for the orthotopic tumors, which were placed directly into the cortex, it was about 50 to 65 milligram. But this is like a bulk tumor.
I asked that mostly because when talking about the macrophages in your work, I thought that if there's a way to differentiate that from the total tissue, maybe something very cool or something like that.
Yeah, so yeah, exactly. But I mean, it's usually, so when because we would try to translate what we see in the biopsy into the imaging, right? You will not separate in vivo in the brain. You will not, if you do imaging, right? So you really would like to know how the total metabolism is different between pseudo progression and true progression. But the data which I showed with the substrates, those were separate cell types, right? So we just use macrophages, and we saw that glutamine is increased in macrophages and only glutamine. And then we use different brain tumor cells, just ex vivo, just in vitro. And we saw that they have also tyrosine, methionine, and glycine increase, and glutamine.
So I have a quick question, Natalie.
Thanks, yeah.
Thank you.
Sorry. Can you hear me?
Yeah. Yes.
So I have a quick question. So if you were, so this is really cool, and obviously the idea is that you're going to be able to hopefully eventually translate this in a way that you can predict whether you've got pseudo progression or actual progression, right? And so as you gear up to do that, what do you do? Do you do sort of large scale animal studies where you do your imaging and then you sack the mice right at that time and then analyze whether you've properly predicted what's going on? And how do you get this from here into like actually being clinically applicable, I guess is my question.
Yeah, so you just have a number of animals and you just irradiate them. And you see with the MRI, just to start with, where you have a higher level of inflammation, where you have lower level of inflammation. And then, as you said, you just sacrifice those animals and do metabolic profiling, and then you correlate metabolic pathways from the ex vivo with the in vivo inflammatory evidence in MRI.
So is that where you guys are moving next?
Yeah, so we're actually doing, yes, it's pretty much it's what we're doing also like in head and neck, because head and neck has exactly the same problem with the pseudo progression. Yeah, it's what we're doing now.
And I've got one last question, although I could kind of ask you this offline. Have you guys published this? This is a pretty cool new innovative method. I'm thinking it would be great for our cancer center renewal.
So we published the differences in amino acids and GBM and normal astrocytes. It has been published already. We also published iron oxide in tumor-associated macrophages, but this particular work is not published yet. No.
Okay. Thanks, Natalie.
Thank you.
Great, and I think we've just hit the 20-minute mark, so we should move on. And before I introduce the next speaker, just to remind everyone that the goal of these is to promote interaction. So please take your conversations offline and make connections from all this. And as I mentioned yesterday, we want to know about it. So tell me about it, tell your program leaders about it. So if there's any positive outcome, let us know. If anything negative comes of all this, we really don't want to know about it. That was a joke. Okay, so now we're moving on to the next speaker. It's Dr. Eric Pietras, who's in the Division of Hematology in the Department of Medicine. And he's going to talk about pseudohypoxia and aberrant glycolysis as the driver of TET2-mediated clonal hematopoiesis of indeterminate potential. All right, Eric, you can take it away.
All right, thanks a lot, James, and thanks for inviting me to share some unpublished work that actually I've not yet presented, so this is kind of a first. So the lab is really interested in a couple of key questions. And when I first started the lab, we were mostly interested in how inflammation impacted the functionality of blood-forming hematopoietic stem cells. But as time went by, we became much more interested in the question of how do we actually get blood cancer? And particularly, what are the earliest initiating steps in leukemogenesis? So the way we currently think about this is that over a lifetime, you have a small pool of hematopoietic stem cells. These are self-renewing cells that give rise to every cell of the blood system. And eventually, somewhere usually within our 40s and certainly by the time we hit our 60s and get older, we begin to exhibit a phenotype known as clonal hematopoiesis of indeterminate potential, or CHIP. It's also known as age-related clonal hematopoiesis, or ARCH. You may have seen it termed that way. And what ARCH simply is basically the clonal expansion of hematopoietic stem and progenitor cells that carry an oncogenic mutation. Now, these cells are not themselves cancer. They require additional oncogenic hits in order to occur or to generate leukemia, but this becomes highly prevalent with age and does increase the risk for development of blood cancer. Interestingly, it also increases the risk for cardiovascular disease and potentially tissue aging in other interesting ways. Now, again, this is largely an aging-related phenotype. It's also associated with bone marrow damage that can be induced by chemotherapy and other injuries. So what we suspect is that probably inflammation and bone marrow damage are actually key drivers of this phenotype. So this brings to mind work from the moderator of this seminar, James D. Grigori, who's spent quite a bit of time developing a theory of adaptive oncogenesis to which we in the lab owe an intellectual debt. Basically, what the theory stipulates is that wild-type cells in a healthy tissue have a high level of fitness, and so it takes a significant amount of sort of uphill pushing to get them to a malignant genotype to actually expand clonally. However, a change in the ecology of the tissue, such as inflammation, can create a very different fitness picture, whereas the wild-type genotype is now greatly disadvantaged. And so this creates a paradigm in which therapy that can target the mechanism that reduces the fitness of the wild-type cells could potentially be a way to restore fitness to wild-type cells and restore tissue fitness in a way that limits the possibility to progress to cancer. Now, in the lab, we use a number of different systems to study this. The one I'll discuss today is actually a common mutation that is found in individuals with clonal hematopoiesis, and that is a loss of function of TET2. TET2 is a dioxygenase. Basically what this means is it's an oxygen-dependent enzyme. What it does is it catalyzes the hydroxylation of methylcytosine. This allows methylcytosine to be, or the methylation group to be removed, and for DNA to essentially now become transcriptionally active. In the context of TET2 deficiency, this leads to essentially a hypermethylated state, and indeed many leukemias are hypermethylated. This is why azacitidine works as a backbone for a number of different therapies, including venetoclax. What we do know, however, is that when cells acquire a loss of function mutation in TET2, and specifically hematopoietic stem and progenitor cells, this creates a sort of self-renewal phenotype where these cells are now able to clonally expand in a much more easy fashion. In addition, they also carry a pro-inflammatory phenotype that has become the subject of significant interest in my lab and others. So the first question we wanted to address was basically to develop a model where we could robustly study clonal hematopoiesis in an in vivo setting. Humans are obviously a little bit problematic to do that. Mice historically are problematic because most approaches have used radiation in order to condition animals to generate the sort of chimeric picture. We recently published work with Ken Walsh down at University of Virginia, in which we used a completely unconditioned system that was actually first developed in the 90s. Basically what you do is you just inject mice with bone marrow. Eventually you'll get a small amount of chimerism in the animal. And what you see is that TET2 deficient cells, whether they are full knockouts in this case, or whether they're heterozygotes, which more resembles the human condition, you see that there's a robust clonal expansion of these cells, or I can't say clonal because we're not putting in a single cell, but simply that there's a selective expansion of TET2 deficient hematopoiesis within those mice. We can detect that in the blood. And then if we look in the bone marrow, we can also see an expansion of hematopoietic stem cells, as well as the downstream progenitors that give rise to the myeloid and lymphoid lineages. In fact, this is primarily where the expansion occurs, which leads us to believe that these metabolically active cells, the MPPs, the multipotent progenitors, and the myeloid progenitors, are really where clonal hematopoiesis is occurring. So what does this mean physiologically? In this particular study, Ken Walsh's group was interested in the inflammatory features of macrophages that have TET2 mutation. And what we can find is that the macrophages produced by these mutated hematopoietic stem cells are hyperinflammatory. They produce high amounts of cytokines, including IL-1 and IL-6. And by lodging in an otherwise normal heart, they can actually thicken the arterial walls and cause the predecessor to cardiovascular disease. Now, in the lab, we were interested in where else these cells disseminate. We actually took a look at a number of tissues that are either hematopoietic or non-hematopoietic for that matter. And what we can find is that after about 8 to 16 weeks, TET2 deficient hematopoiesis spreads throughout every tissue in the body that is otherwise home to tissue resident macrophages and immune cells. In fact, when we look at multipotent progenitors that are TET2 deficient, these end up homing into those tissues, now providing ready sources of mature blood cells that could potentially contribute to tissue dysfunction, aging, and even carcinogenesis. So we think this is a mechanism that's potentially applicable to studying a number of different cancer models, and we're always looking for collaborations along this line. In order to develop a little bit more of a reductionist system, what we did is just look in a simple culture condition as to whether or not we could replicate some of these key features. Here what we did is we simply cultured c-KIT positive HSPCs, which is kind of equivalent to human CD34 positive cells in liquid media, and we grew them in an incubator for a period of 12 days, and indeed we can see in the red the expansion of the TET2 deficient hematopoietic stem and progenitor cells. They eventually become myeloid cells in this condition. Collaboration with Ravi Majeti over at Stanford shows that using a CRISPR system, we can do precisely the same thing in human CD34 positive HSPCs. Now, one thing that was interesting is This phenotype is, as it turns out, is oxygen powered. And this actually makes a lot of sense for two reasons. One, the bone marrow under normal conditions is quite hypoxic. It's PPO2, or partial pressure of oxygen, ranges from about 1% to maybe 3 to 5% in certain areas. And on top of that, TET2 is a dioxygenase, meaning if there's no oxygen around, it's inactive. So we think that's part of the collaboration between the bone marrow environment as a hypoxic environment that limits the anti-sulfur-renewal effects of TET2, and at the same time, basically helps promote stem cell quiescence and long-term function. But one thing we noticed is that when we culture these TET2 deficient cells in very oxygen-starved conditions, 1% oxygen, we actually don't see a substantial amount of expansion of those cells over wild type. In fact, almost nothing grows, so it actually looks a lot like healthy bone marrow. But as soon as we start increasing oxygen content out to normoxia, what we find is that this is actually quite a different picture, and now we begin to see the selective expansion of TET2 deficient cells. So given that we wanted to address molecular mechanism, what we did is we ran RNA-seq on hematopoietic stem and progenitor cells that were simply cultured for about 12 hours to initiate whatever molecular program we were trying to study. And what came out of our RNA-seq study was actually two key gene programs here. One is glycolysis, the other hypoxia. These were both the highest scoring gene programs within the overrepresentation analysis that we conducted. And in fact, what we could see is that a whole gamut of glycolysis genes were strongly upregulated in TET2 deficient HSPCs. When you lay them all out kind of in a linear way with the pathway, you can see that everything from glucose transporters such as GLUT1 through processing to develop lactate and also to secrete lactate, MCT4, all were upregulated, suggesting that we were potentially looking at the making of a Warburg shift, or basically an aerobic glycolytic phenotype. So we collaborated with Angela D'Alessandro's lab and ran metabolomics, kind of an initial screen that we have to come back to on these cells. And what we noticed is that, very similar to the gene expression data that we saw, we actually get a substantial increase in all of the ingredients through the glycolytic pathway, including NAD+, which is something that has to be continually cycled in aerobic glycolysis. We didn't see substantial increase in pyruvate. We think that could be in part because whatever pyruvate is made actually gets used a little bit more efficiently. We do see increased oxygen consumption just based on seahorse analysis. So what we think is that there's increased glycolysis. Its end products can potentially either be shunted to oxphos for ATP generation or to lactate in order for ATP generation to occur in that setting. And indeed, this glycolytic pathway seems to be crucial for maintaining stemness in the hematopoietic system. So altogether, it looks like these cells are better at generating energy. So what's the mechanism underlying that? The first thing we turned to was HIF-1 alpha. So HIF is of course responsible for activating a large number of these glycolytic genes, and that basically drives the glucose in, lactate out sort of pathway that is often dubbed pseudohypoxia. In other words, activation of hypoxic programs in the presence of oxygen, which is exactly what we see. Now, we don't see changes in HIF-1 alpha at the mRNA level, but work from Gang Huang over at Cincinnati has shown that TET2 deficient cells have more HIF-1 alpha protein. And this is important because HIF-1 alpha is regulated very significantly at the post-translational level. In other words, its stability governs its activity. It's often ubiquitinated as soon as you get into sensing oxygen. So we decided to look at three potential mechanisms that could be driving regulation of HIF activity in this context. One is simply repression of genes that promote the degradation. We didn't find any of those genes repressed. In fact, we find a couple of these PHD proteins are actually increased in expression at the mRNA level. We don't yet see any evidence of metabolites such as succinate that stabilize HIF-1 alpha. We have to have a closer look, but we're not necessarily sure that's the picture. On the other hand, inflammation has been shown to stabilize HIF-1 alpha. In fact, inflammation through cytokines such as IL-1 or IL-6 are well known to induce stabilization of HIF-1 alpha. So it's important to note that work from Ruben Kapoor's lab has shown that, and as well as work from our collaborator Ken Walsh, has shown that TET2 deficiency leads to a licensing for overproduction of IL-6. And so we think this could potentially be the inflammatory lever that helps to stabilize HIF. Indeed, there's work showing that STAT3, which is directly downstream of IL-6, is able to prevent the degradation of HIF-1 alpha by essentially covering up the site that proteins like VHL would normally bind to. So collectively, what this gives us is an emerging picture that we're currently in the midst of trying to test to see if we can develop potential therapies that can eradicate these cells. So it's well known that IL-6 production is licensed in TET2 deficient hematopoiesis because of the fact that TET2 is absent and therefore, or is mutated and therefore can't recruit HDACs to suppress IL-6 production. So we think that IL-6 produced both by TET2 deficient cells themselves, as well as by damaged or aging bone marrow, can exacerbate a HIF-1 alpha phenotype in these cells. And that together drives the pseudohypoxic phenotype that we're observing here. We think this cooperates with and may well contribute to the aberrant self-renewal gene program that TET2 deficient cells have, thereby partnering with other molecular deregulations to drive clonal hematopoiesis. What this also outlines for us is a series of key targets that we need to investigate in terms of their ability to both stop this pseudohypoxic program, as well as to eradicate or at least limit the expansion of TET2 deficient HSPCs. And that includes IL-6 and STAT3, HIF-1 alpha itself, and other constituents of the glycolysis pathway. Indeed, along those lines, we've made initial examinations at blocking MCT4, which is the monocarboxylate transporter that exports lactate. Otherwise, lactate becomes highly toxic in a cell. And what we find is that monocytes and indeed myeloid cells in general tend to express this. So this could actually be relevant to clonal hematopoiesis. So what we did is we took a cue from a previous report from 2016 and treated ourselves with serosingapine, which blocks MCT4 and MCT1, and metformin, which basically helps prevent OXPHOS from regenerating NAD+. What we find is at least a reasonably modest sort of inhibition of TET2 deficient clonal expansion. So we think this could be at least part of a mechanism to try to prevent CHIP before it starts. We're also interested because MCT4 is so highly expressed in monocytes and macrophages, could it even just selectively deplete the hyperinflammatory macrophages that cause heart disease and may contribute to cancer? So in conclusion, TET2 deficient HSPCs exhibit selective growth advantage. This is largely, we think, through activation of a pseudohypoxia gene program that exacerbates glycolytic ATP production. We think aberrant HIF regulation is the link between hyperinflammation and pre-leukemia in this context, and we believe that IL-6, STAT3, and glycolysis become key targets for us to investigate. Where this sits in terms of putting together our pilot grant is currently we're very interested in two questions.
And Eric, go quick so you have some time for questions.
Perfect. Yep, we're just about done. IL-6 and STAT3 drive pseudohypoxia in TET2 deficient HSPCs, and does blockade of the mechanism prevent the expansion? And then the broader question is how common this is, and can it also eliminate tumor resident cells? So I think the overall implication is basically that we see pre-leukemia as sort of a triangle, and if you pull away any one of these arms, kind of like a fire triangle that we learned about in grade school, you can potentially collapse the system. And we think this could help to basically prevent disease at early stages, such as CHIP, before it becomes malignant. And so lastly, thanking members of my lab, as well as our collaborators here and collaborators elsewhere whose work has been instrumental for us to help to understand our system. Thank you.
Great, thanks, Eric. That's just awesome. Questions? We've got time for a few questions.
Eric, Sid Mitra here from Pediatrics again. That was fantastic. I have one question and one word for you.
Yeah.
The word is parabiosis. And the question is, do you know if there is an age-related component to the expansion of the TET2 knockout cells?
Yeah, Sid, very good question. So I'll get to the second first, and probably the word we should discuss offline, because it is an interesting point, and a number of parabiosis experiments have been done between heterochronic animals for hematopoietic aging. But to the issue of and sorry, just remind me, I actually talked my way through remembering what your second part of your question was.
Is there an age-related component to this expansion that you see from the TET2 knockout, TET2 deficient? Sorry.
Yeah, so, certainly in terms of the statistics as to when people actually get the clonal expansion. So in humans, we can detect these clones in rare form in 95% of people who are age 40. By the time you get to age 80, the prevalence of individuals with higher allele frequencies is increased. Have we modeled this in the mouse rigorously? No. And that's in large part just some limitations of the system that we're actually hoping to investigate. I think the emerging answer in the field is that absolutely there is some sort of age-related environmental context that promotes this sort of expansion. We don't know exactly what the drivers are, but I anticipate they're going to be inflammatory cytokines and potentially bone marrow structure disruption.
I think that's exactly where the parabiosis comes in.
Mm-hmm.
Get that by Io Cook.
yeah, that'll be a fun one to get past. But we've got some collaborators, so we'll see what we can do.
No, I think with the aging component, it's going to be important to start with very low percentages of TET2 mutant. I mean, that's what we've always seen in our experiments with the aging models. So even when you do that transplant, I think that there's a self-feeding with the TET2 mutant cells or with their own inflammatory cytokines may obviate the need for aging in that model. But if that percent was really low to begin with, you might be able to see that expansion because essentially it wouldn't be able to feed itself.
Absolutely agree. And I think that's the beauty of the system we're using, because essentially we're starting with a VAF of TET2 deficiency that's somewhere around 1 to 3%. So it's rare, and we can watch it expand in a native environment that has been completely unmanipulated. So we can actually do the manipulations and don't have to worry about radiation or chemo or any of these other things that actually cause the clonal expansions to begin with. So everybody else has been kind of cheating. They put the cells into an irradiated background, and of course they're going to expand. It's an irradiated mouse at that point.
Yeah, cool. All right, well, we're going to need to move on to the next talk. Thanks a lot, Eric.
Okay.
So the final talk is from Dr. Haobin Yi. He's a postdoctoral fellow in the Jordan Lab, and he's going to be talking about the hepatic microenvironment uniquely protects leukemia cells through induction of growth and survival pathways mediated by LIPG. So take it away, Haobin.
So when we talk about microenvironment for leukemia cells, we always think about the microenvironments reside in the hematopoietic active cells such as bone marrow. But due to the nature of the disease, leukemia cells could possibly invade multiple types of the cells. So therefore, there are multiple types of microenvironment for leukemia cells. For instance, our recent study has demonstrated that leukemia cells could actually invade adipose tissue, and interactions between adipose tissue and leukemia cells reprograms the resident leukemia cells and create a distinct population that are quite resistant to chemotherapy. So in another recent study using PET-CT scanning technology, we found that the liver tissue in leukemia mice is heavily infiltrated with leukemia cells. In addition, it has been reported that hepatic infiltration of leukemia cells has been in up to 75% of acute myeloid leukemia patients. So these information suggests that liver might function as a reservoir for leukemia cells. So we hypothesized that there is a hepatic microenvironment for leukemia cells. So in this project, we used 3 murine models, and all these 3 murine models give us similar results. And we also have PDX model and patient samples to demonstrate what we found in murine models are also true in human patients. But because of the time limitation today, I'm going to only show you the results we get from the first model. So in this model, we transform the normal hematopoietic X cells with 2 leukemic oncogenes. And these 2 leukemic oncogenes were tagged by GFP and YFP respectively. And then we then transplant these transformed cells into recipient mice, and we can use flow cytometry to detect the presence of leukemic cells as shown by the GFP and YFP double positive population. So using this mouse model, we found that the liver tissue in the leukemia mice is heavily infiltrated with leukemia cells. And we also see severe liver damage in leukemia mice, and we'll come to this point later. So when we compare the leukemia burden between bone marrow and liver, the liver actually has a more leukemia burden compared to bone marrow, suggesting that the hepatic microenvironment promotes the proliferation of leukemia cells. And indeed, in vivo BRDO incorporation assay confirmed that the liver leukemia cells have a higher incorporation rate of BRDU. So these data suggest that the hepatic microenvironment promotes the proliferation of leukemia cells. So to further characterize these leukemia cells in the liver, we performed transcriptome analysis and found, compared to bone marrow leukemia cells, liver leukemia cells display a distinct gene signature. And we found these pathways were highly enriched in the liver leukemia cells. And 2 of these pathways were related to cell cycle. So again, suggests that the liver microenvironment promotes the cycling of leukemia cells. So then we asked, what's the mechanism underlying this increased cycling status of liver leukemia cells? So since liver is a metabolically distinct and active tissue, we want to solve this problem from a metabolic perspective. We compare the metabolome of liver and bone marrow leukemia cells and found one class of metabolites are highly enriched in liver leukemia cells. And this class of metabolites is polyunsaturated fatty acid, or PUFAs. So next, we examine the mitogenic effects of these different species of PUFAs in vitro and found the linoleic acid, or LA, is the most mitogenic PUFAs amongst these test PUFAs. And when we treat leukemia mice with LA, it promotes the engraftment of leukemia cells in bone marrow and liver. Additionally, we found that the LA promotes the progression, promotes the proliferation of leukemia cells through activating ERK signaling pathway. And not surprisingly, we found the ERK signaling pathway is more active in the liver leukemia cells compared to bone marrow leukemia cells. And when we treat leukemia mice with an ERK inhibitor, it selectively blocks the engraftment of leukemia cells in the liver. So next, we asked, what's the mechanism underlying this elevated level of LA in leukemia cells that reside in the liver tissue? So we went back to our gene analysis data and found one pathway called glycolipid metabolism pathway is highly enriched in liver leukemia cells. And the top dysregulated gene in this pathway is called LIPG. So what is LIPG? LIPG is a lipase that could convert the phospholipids in HDL to fatty acid and therefore promotes the turnover rate of HDL. So then we ask whether this LIPG could regulate the cellular level of LA. So first, we overexpress this gene and found the LA is the only species of PUFAs that are upregulated by the overexpression of LIPG. And on the contrary, when we knock out this gene in leukemia cells, the cellular level of linoleic acid is decreased. So together, these data suggest that the LIPG promotes the accumulation of LA in leukemia cells. So we also found when we overexpress LIPG in leukemia cells, it activates the ERK signaling pathway, and with the presence of HDL, this activation is even stronger. And when we transplanted these overexpressed cells into mice, the engraftment of these cells are much better compared to control cells. On the contrary, when we knock out this gene in leukemia cells, the ERK signaling pathway activation is decreased. And when we transplanted these knockout cells into recipient mice, the leukemia burden in the liver is decreased compared to control cells. So together, these data suggest that LIPG promotes the proliferation of leukemia cells. So one phenomenon we observed during this project is that the the viability of LIPG overexpressed cells is always better than control cells, suggesting that LIPG might promote the survival of leukemia cells. And in another set of experiment, when we treat leukemia mice with chemotherapy, the residual leukemia burden in the liver is always higher than that in the bone marrow, suggesting that the leukemia cells reside in the liver is chemoresistant. So then we think whether there is causal relationship between LIPG expression and the chemoresistant of liver resident leukemia cells. So what we did is we transplanted these overexpressed into recipient mice and then treat these leukemias with chemotherapy, and then examine the residual leukemia burden after chemotherapy. And indeed, we found that the leukemia burden is much higher in the mice transplanted with overexpressed cells after chemotherapy. And on the contrary, when we transplanted knockout cells into recipient mice and then treat these mice with chemotherapy, the residual leukemia burden is decreased compared to is decreased in the mice transplanted with knockout cells compared to mice transplanted with control cells after chemotherapy. So together, these data suggest that the LIPG promotes the survival of leukemia cells upon chemotherapy challenge. So next, we ask, what's the mechanism underlying this LIPG-mediated survival of leukemia cells? So we tested several hypotheses, and long story short, we found that LIPG could promote the expression of BCL2 and BCL-XL. As you can tell from this, these Western blot, the overexpressed cells have a higher expression of BCL2 and BCL-XL protein levels. And in the knockout cells, the BCL2 and BCL-XL protein levels is decreased. And we also saw that the liver leukemia cells have a high expression of BCL2 and BCL-XL protein levels. So these results give us a hint on how to target these leukemia cells in the liver tissue. So we combined chemotherapy with ABT263, which is an inhibitor for BCL2 and BCL-XL. And we found the combination significantly kills the cells reside in the liver tissue, while this combination has minimal effect on the bone marrow leukemia cells compared to chemotherapy alone. More importantly, this combination therapy promotes the survival of leukemia cells. So when I mentioned earlier that the leukemia disease could cause liver damage, and indeed we found the serum activity of AST and ALT is increased in leukemia mice, suggesting indeed there are liver damage in leukemia mice. So liver is the main tissue to detoxify chemotherapeutic drugs. So one enzyme called CDA could convert AREC to AREU. AREC is a chemotherapeutic drugs that are commonly used for leukemia patients. And after converted by CDA to AREU, the toxicity of AREC is decreased. And we found the serum activity of CDA is increased in leukemia mice. So these data suggest that the liver damage caused by the disease could cause release of the chemo drug degrading enzyme to the peripheral tissue, and therefore protects the leukemia cells in the peripheral tissue from chemotherapy. To further confirm the elevation of CDA activity in leukemia mice, we incubate AREC with either normal or leukemia serum, and then incubate them overnight. And then we measure the AREC amount after incubating. We found the AREC amount in the leukemia serum mixture is a significant decrease compared to normal serum, and the amount of AREU is increased in the leukemia serum mixture. So together, these data suggest that the liver damage caused by the disease leads to the release of the chemo drug degrading enzymes to promote a systemic chemoresistance. So to summarize, what we found is that the hepatic microenvironment selectively increased the expression of LIPG in leukemia cells. And this LIPG has two functions. So one is to promote the proliferation of leukemia cells in the liver tissue. It promotes the proliferation through regulating of PFA metabolism in leukemia cells. And it also promotes the survival of leukemia cells through regulating BCL2 and BCL protein levels. And what I didn't show today is that the LRPG could promote the stability of BCL2 and BCL protein. So therefore promotes the expression of these protein levels. Additionally, leukemia disease could cause liver damage and then leads to the release of chemodrug degrading enzymes to the peripheral tissue and therefore further protect the leukemia cells from chemotherapy. So with that, I'd like to thank my mentor, Dr. Craig Jordan, and all the Jordan Lab members and our collaborators and our funding staff. And thank you for your attention.
Great, thanks. So that was really very cool, Halvin. Let's open the floor up to questions. We actually have quite a bit of time for questions. I'll get one started while we're waiting for others to come up with theirs. So Halvin, the drug that you saw, degradation of ARC, do you think this is going to apply to other chemo drugs, or do you think this is unique to ARC?
So CDA has been the main enzyme for detoxifying ARC is CDA, but CDA can also detoxify other drugs such as doxorubicin. But there are other enzymes that could also degrading doxorubicin or other chemo drugs, but we haven't tested these enzymes yet.
Okay, great, thanks. Harry, I see you're writing in the chat. You want to just ask your questions?
Sure. So it's very interesting to see how the leukemia burden is engrafted in liver a lot higher than bone marrow in your model. Did you also observe the same phenotype in other leukemia models, or like even patient samples?
We do see higher engraftment of leukemia cells in the model with the mutation of FRIT-ITD, DNMT3, and NPM1C. But we see equal engraftment of leukemia cells in the liver tissue in the MLF9 mouse model. Does that answer your question?
Yeah, that sounds like it. Are there any other questions? All right. Well, great. Thanks so much, Alvin. That's really fantastic work. We look forward to seeing it out there.
Thank you, James. Yeah.
And so that's it for the symposium. Once again, thanks. I want to thank all the speakers and for the really great talks. And I hope that some good connections come out of all this. And we look forward to seeing some of your applications for the RFA as well. And there's going to be a sort of an analogous mini symposium, hopefully with the grants program as well, for aging in cancer, kind of in the mid-fall. So look out for that one. All right. And everyone have a good rest of your day. Take care.